Okay, good afternoon. Okay, we're going to start our afternoon series. I just wanted to announce that we have representatives here from 23 states and Canada today. Okay, now Dr. Evinger is going to speak about animal models for blepharospasm and an update on general research for facial spasms. Thank you.
So, animal models in neurological diseases serve at least two critical functions. One, they're useful for testing the safety and the efficacy of new drugs or new treatments of any kind. And secondly, they enable us to identify changes in the brain that could actually produce neurological diseases in a way that's not possible to do in people. And so what I want to talk to you today about are investigations into those possible mechanisms for producing blepharospasm. So let's talk about what we know from the human studies. And this is much of what Dr. Hallett talked about. And he was talking about the phenotype, the spasms of lid closure, and the influence of genetic background, and which was the G, and the environmental trigger, which is the M. And so I want to talk to you today about those two things, two possible mechanisms. And the first, the genetic, as Dr. Hallett pointed out, has been very hard to find. I mean, there haven't been any obvious gene changes, but we think that this genetic background, whatever the genes are that are involved in it, what they do is they create low-frequency brain oscillations which exaggerate motor learning. And when an environmental trigger occurs in that environment, motor learning gets exaggerated and leads to spasms of blepharospasm. Now, I'm going to spend most of the time talking about these low-frequency brain oscillations. And actually, they sound strange, but they're not as strange as you might think. But let's start by talking about what we know about the environmental trigger. Now, we've hypothesized that the environmental trigger is dry eye irritation, ocular. Now that's hardly an idea that's original to me. Lots of people have done it. And out of the human literature, this is a study from Dr. DeFazio's lab, which says that there's a significant association between ocular symptoms, they're talking about dry eye, that sort of thing, and BSP, which is abbreviation for blepharospasm. And so we want to go back to what dry eye is, and we're going to go back to the trigeminal system, and I'm going to repeat some of the things I said before, hopefully sometimes a little bit more clearly this time. And so that is, remember, the trigeminal system is how information from your cornea gets into your brain through the trigeminal system. It comes in through the trigeminal nerve into the trigeminal nucleus. And when your cornea gets exposed and you get lots of pain signals coming to the trigeminal nucleus, you get sensitization, and that as you undergo motor learning, which changes your blink circuit so that you can start compensating for the dry eye. Keep the tear film as going as well as you can. All right? Now, just like we said before, here's an eyelid, goes down, comes back up. We're stimulating the supraorbital nerve, which is that nerve right there. And normally you get a single blink. As I said before, when you have dry eye, you get multiple blinks. These are data from a dry eye patient. You also get an increase in excitability. Remember I talked about how you want to suppress a blink after you've made a blink so you don't get blink after blink after blink. And this is all done through the trigeminal system. And when you have dry eye, you get more excitability than normal. We measure this test by condition ratio This would be 0.1 for control for this person before they had dry eye, and 30 minutes later after they got eye irritation. Now this is value of 0.06. So dry eye makes reflex blinks more excitable, and you cause lots of additional blinks. All those things that you would expect to compensate for dry eye. They help with dry eye. And the two are linked. Right? What's plotted here are the number of additional blinks you make as a function of how excitable you are. If you're more excitable you are, we're using that test condition ratio, the higher this number, and the more additional blinks like this. And you can see you get a tight relationship there. So these two are linked, and they are learned behaviors. They're motor learning. Okay? They're not a reflex. They're something you learn. Okay? Your brain changes. It adapts its plasticity, like Dr. Hallett was talking about earlier. So let's think about what those, I've shown you all these graphs. What's it really look like? So here's a kid with eye irritation. Okay? He doesn't have blepharospasm. He has eye irritation. Okay?
Will he get blepharospasm?
I don't think that's unlikely. Okay? He's very young. I mean, think about it this way. Dr. Stefko can tell you more than I could, but I think dry eye's got to be probably the single most common ophthalmological disorder that people go through. And certainly as you get older, lots of people get dry eye, but as you know, very few people get blepharospasm. Okay? And the reason for that is this whole issue of blepharospasm, I would argue, is an exaggeration of these symptoms. I mean, if you think of exaggerating what you saw that little boy doing, it'd be like blepharospasm. Right? So how do you go about exaggerating that? And why do some people develop blepharospasm and most people don't? Well, we're back to this issue of the influence of a genetic background. Okay? And what is that background? I think that that's what's happening is you're amplifying low-frequency oscillations, and that these oscillations exaggerate motor learning. All right, now you're saying, keep saying low frequency brain oscillation. What in the is he talking about? I mean, this is just. Let me explain it to you. It's actually, in some ways, fairly simple. So let's think for a minute. What you have here, this is a recording from a monkey, and these are 3 different neurons, all recorded at the same time. And this is in a part of the brain called the basal ganglia. Okay? And so you're seeing the electrical activity. Now, all your neurons, every neuron in your brain works by electrical activity. And they communicate with each other through electrical activity. And the communication that they're using are these, what you see lines here, are called spikes or action potential. And every time one of those things occurs, it communicates something to another neuron. Right? Now, if you look at this, most of these neurons aren't synchronized. That is, they aren't doing the same thing at the same time. And the easiest way, in many ways, to think about the brain, because the brain is composed of, billions of neurons. Right? And what are they all doing? Are they all doing the same thing at the same time? What are they doing? The easiest way to think about it is the crowds, a crowd of people. Right? So if you think about it, there's not much synchronization. Now, here's a group of people just walking down the street. Okay? Most of the people, they're unsynchronized. They're not doing things. Here's a group that are together. There's a couple walking, a little bit of synchronization here and there. And things are getting done. Right? Your brain is going through, things are getting done because everybody's little groups are synchronized and they're taking care of things, and other groups are just moving along doing other things. But everything's getting done. People walking down the street get a lot accomplished, right? They go into stores, all sorts of things like this. Now let's think about what happens when the brain gets synchronized, as it does in disease states. This is an example from a monkey again, who has the monkey equivalent of Parkinson's disease. And again, here are 3 neurons, okay? And now you can see what they're doing is these action potentials occurring in bursts. Like instead of going no, 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 like this, they're going. Okay? They're going in bursts. Now, these bursts turn out to be synchronized among the neurons. So instead, like up here, neurons really weren't doing that much with each other. Now, in the disease states as Parkinson's disease and dystonia and blepharospasm, a lot of the neurons start synchronized with their neighbors. Synchronization can be a very bad thing. Think of your crowds again. And here's an example of people outside Urban Outfitters at 5 a.m. on Black Friday. Right? There you go. They gummed up the works, right? You get too synchronized, things are not good. All right. Now, how do you see this in people? All right. Now, oh, sorry. Skipped one over one here. Now, how you can see this in people. So, okay, let's see. Try and get over here. So none of you are going to, allow people to just go stick electrodes in your brain and record. Okay? Or if you're reasonable, you should not allow anyone to do that to you. Okay? But as Dr. Richardson is going to talk to you about, you can put a series of electrodes in the brain to do deep brain stimulation, and it alleviates disorders. And I'll talk a little bit more about deep brain stimulation. But when you put those electrodes in the brain to electrically stimulate, to activate neurons, you can actually also use them to record neurons. And you can't record individual neurons very well, but you can record the activity of lots and lots of neurons. And that's what's happening right here. So here you can see, this is from a patient with Parkinson's disease. And you can see this is one neuron giving these bursts, and you can see that it fits with this slow activity here. And these are local field potentials, and when you see those, what that represents is thousands of neurons all doing the same thing at the same time. If they were all doing different things, that line would just be sort of wavy flat. Right? But when they're all doing the same thing, when they're all going bump, 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 bump together, then you get these oscillations. Okay? So that's how we know it's going on in people. Now, the frequency at which they do it is going to turn out to be important. Okay, so let's look here. Now, here what you have is, this is a Parkinson's patient. Here's 1 second, and you can see these local field potentials going like this. Now, the way we measure those, if you think about this wave right here, all right, we'd say 1 cycle would be like from trough to trough. That would be 1 cycle, okay? And you can see that in this 1 second period, we had 3 complete waves. Okay? So we'd say these are going at 3 cycles per second, or 3 hertz. We go into the technical stuff. But this neuron, or this group of oscillations, are going much faster. They have a shorter, it takes less time to do 1 cycle. And so there's 6 of these in each second, so you have 6 cycles per second. Now, the frequency issue here is, let's look at the Parkinson's patient. Here are these oscillations going on right here, recorded through deep brain stimulating electrodes. Okay? And it turns out that they're oscillating somewhere in the range of 16 to 20 times every second. So a patient with Parkinson's disease, instead of having this sort of, unsynchronized activity, is having this synchronized activity in every, 16, 17 times a second, they're all giving these little bursts. Parkinson's disease is a classic example, right, of the works being gummed up. Because what's the definition of Parkinson's there? It's hard to move, it's slow to move, that sort of thing. So you can think about these oscillations coming up the works. Now let's look at dystonia. And so this particular example is from a patient with generalized dystonia, the worst kind of dystonia. And they're recording from them. And the thing that's most striking is these waves are much slower, right? They occur much less frequently than this. It's like comparing this 3 cycles per second to this one being Parkinson's and this one being dystonia. And they come out in a different frequency range. This is sort of the frequency at which they're oscillating. And you can see more low frequencies in dystonia and slightly higher frequencies in Parkinson's disease. So the exaggerated oscillations in the basal ganglia, which is the part of the brain that we think is most involved in these sorts of things, is much higher in Parkinson's disease than it is in dystonia, of which blepharospasm is a form of dystonia. All right. What I've been arguing is that the genetic background amplifies these low frequencies, these ones less than around 7 times per second, as you see in dystonia, and exaggerates motor learning. And you're saying, wait a minute, you just told me that synchronized oscillations gum up the works, right? So synchronization can't be good. Synchronization is not entirely bad. I mean, here's an example, right? This is from the World Cup. Right? A wave is synchronized activity. Now, things that does some things very well, although if you're trying to watch the game, it's a nightmare, right? But it does some things very well. And what we think is it's exaggerating motor learning. And so this is where the animal models come in. So all these data I was showing you about the oscillations have mostly come from people, from recordings from people with these deep brain stimulating electrodes. And so you have to ask yourself, are these oscillations sufficient to exaggerate motor learning? Can you exaggerate motor learning with them? And we played around with a lot of ways. How can you show this? How can you do this? And then I said, well, let's take a normal animal, in this case a rat, and we're going to put an electrode into the part of the basal ganglia called the subthalamic nucleus right here. This is where the electrode was. This is a section through the brain as if you were looking like this, pulled away and looked straight in. This would be the way the brain would look. And we put an electrode there and we did stimulation. Okay? Deep brain stimulation. And we stimulated at different frequencies. We stimulated at 7 hertz, which is a frequency associated with dystonia. We stimulated in the same rat at a different time, not all at the same time, 16 hertz, which is typical of Parkinson's disease. And then we did 130 hertz. You're saying, 130 hertz? Where did this come from? It turns out, and I'm sure Dr. Richardson will talk more about that, when they take patients and use deep brain stimulation, they stimulate at 130 hertz. Now, I have no idea why that number was picked, but it works exceptionally well. Okay? So now in a normal rat, you'd have a certain set of predictions. Okay. If we're right about this, when I stimulate these normal rats at 7 hertz, just continuously, they're getting 7 hertz stimulation all the time, that motor learning would be exaggerated. Okay? On the other hand, in Parkinson's disease, motor learning is impaired. And so the argument would be if these oscillations really make a difference, if I'm stimulating at 16 hertz, you should be impaired learning. And then 130 hertz, I'm one behind, aren't I? And then at 130 hertz, you shouldn't get any effect at all. Okay? So we're testing these 3 different frequencies, all within the same rat. Okay, so we've got a normal rat. All right, so let's look at a form of motor learning. Now, this is a system that we designed in humans in which we would measure blink amplitude before, and then we would do a high-frequency stimulation during the blink, and we could either, depending on the way we did it, we could make the effect would be to potentiate blinks, make them larger, or to depress them to make them smaller. So and then afterwards, we'd look immediately, 30 minutes and 60 minutes afterwards. So here's a normal subject. Okay? Now, this is the EMG activity of the lid closing muscle. This is the one that Dr. Hallett showed you. All right? And so this is the baseline. That's before anything happens. Then after you do this high-frequency stimulation, if you look immediately after 30 minutes or 60 minutes, you can see that this blink is larger, right? Everybody would buy that. Here's a Parkinson's patient. Start out with a baseline, go through this exact same training paradigm as this person, and what you can see is there's no change in the blink amplitude. That learning is impaired with the 16 hertz. All right. So let's take a rat now, a normal rat, and by control we mean we're not stimulating. And in this case, our goal was not to increase the amplitude, but to decrease the amplitude of the blink. So when you take this rat without any deep brain stimulation, no stimulation at all, here's before, here's a T0, and here's a T6. So you can see the blinks are getting smaller. If we do the same experiment on the same rat, except now we're doing 16 hertz, which should be like Parkinson's disease, okay, you don't see any change in the amplitude at all. Learning's impaired. And you can quantify it this way. So here's where you're trying to potentiate the blink reflex. Here's that 0, 30, and 60 minutes after. Here you can see a control subject, and you can see that their blinks, 100 would be what it was, the baseline. So they're, 20 to 30% larger than they were before we did this training paradigm. 20 to 30%. If you look at a Parkinson's patient, okay, they are just showing a tiny, tiny increase, which is not really significant. Okay? Here's what happens when you look at the rats. So no DBS, and now in this time we're turning the gain down rather than up. That's why the bars go down instead of up. There it is. You can see that the gain goes down like this, about 25%. If you do 130 hertz, right, that's the therapeutic. It shouldn't have any effect on this at all. And indeed it doesn't. These rats show just about the same amount of reduction in blink amplitude. But when you do 16 hertz, you can see there's virtually no change. And when you do a 6-hydroxy lesion, which is sort of a rat model of Parkinson's disease, you get the same ability. So the 16 hertz impairs motor learning. Now the question is, what happens when you do 7 hertz? Right? Because that's dystonia. I'm arguing that's going to exaggerate motor learning. And that's exactly what it does. Okay? You get an increase. You get more depression of blinking than you do normally, and these are normal rats. And when you compare that with blepharospasm patients, all right, so here are normal patients in that same paradigm, except this time is to increase the amplitude of the blink. At 0, 30, and 60 minutes, you can see that control subjects are, somewhere between 120, around 120 overall percent change. They're 20% larger. Look at the blepharospasm patients. They're up 160, 140, 170. They've really got an exaggerated form of learning. Okay? So our argument is that these 7 hertz oscillations allow the changes in blinking that you get with dry eye, this motor learning that occurs with dry eye, to become exaggerated and turn into spasms. Okay? Now, I'm going to show you some really very preliminary data about how this might happen. All right. So let's start with the idea of just giving 7 hertz. And the way we do this, we put the rats, they're in their home cage, wandering around doing whatever rats do in their home cage, and we're delivering this 7 hertz stimulation all the time. It doesn't make them move funny or anything like that. You really can hardly tell that they notice. They really don't seem to appear to notice it's going on. And you'll find the same thing when Dr. Richardson talks about DBS. Patients aren't aware, other than the way they change their movements. It's not like they're aware that they're being stimulated inside their brain at 7 frequencies. Okay? So what happens here is you don't get any spasms of lid closure. What you actually get are standard blinks. So this is, at a very slow time scale, the activity of the orbicularis oculi, that lid closing muscle, which you've heard Dr. Setko and Dr. Hallett both talk about, in a rat. And this is just a spontaneous blink. He's just wandering around his cage, and you can see this blink, and this is expanded. And these are just normal blinks. No big deal. Okay. Now let's look at a rat who just has dry eye. Okay, this is actually the same rat. It's going to show just dry eye. Okay? Now what do you see? You see more frequent blinks, and you actually see him squinting. Right? So there's nothing going on right here. Then you see this blink right here. Right? It's just like these. You can see the expansion of the blink. And you see this activity right here. This is when the rat's sort of squinting, which is sort of typical if you think about when you have eye irritation, dry eye, you blink and squint and all those sorts of things. Now the question is, what happens when we combine the 7 hertz deep brain stimulation all the time with the dry eye condition, this mild dry eye condition? And what you get are these spasms of lid closure. Okay? You don't get it by either one by themselves, but only with the two. And like I said, this is very preliminary to Zeta, but I think what it's sort of trying to show is that we may be honing in on what the genetic changes may actually be doing, what their cause is, what they're causing, and we think that may have to do with changing the way the brain, the neurons talk to each other, so that instead of being mostly unsynchronized, all of a sudden, for some reason, some group of them, particularly ones that may be involved in the eyelids, start synchronizing, going together. And we think that this genetic background may be what causes the exaggerated motor learning of dry eye, which we consider the environmental trigger. So let me finish by thanking lots of people who helped with this study. That's supposed to be well, it is a picture of Stony Brook University, where I come from. They swear it wasn't Photoshopped. To be perfectly honest, I've never seen it look that good. And so in addition to all the people who helped me, I'd also like to thank the Blepharospasm Foundation for inviting me here to speak. I think this is one of the best patient organizations. It is the best patient organization that I've ever been involved with. So thank you very much.